Welcome to the deep dive. Today, we're going to dig into a really fascinating group of organisms. They've kind of baffled scientists, defying easy classification.
Yeah, they've definitely caused some taxonomic headaches.
We're talking about the Archiascomycetes. It's this corner of the fungal kingdom that's, well, they're real mavericks. You'll see why.
Absolutely. And our mission today is basically to take the dense info from Introductory Mycology, specifically the chapter on these guys, and make it make sense.
Right.
We'll look at their structure, how they reproduce, which is sometimes pretty weird, their genetics, and why they actually matter in the real world. You should expect some aha moments. They're surprisingly interesting.
Totally, because it's not just about tiny things under a microscope, right? We're talking about fungi with huge effects. Some are plant parasites causing big problems in farming. Others are super important model organisms for genetic research. And there's even one that, like, for decades, everyone thought was a protozoan, but actually causes serious human disease.
Yeah, it's quite a story.
It really is a wild ride through some seriously adaptable life forms. Okay, so Archiascomycetes. When we first look at them, the big thing is they really mess with how we thought we classified fungi. Yeah.
That's the core of it. They really do.
And the breakthrough was this rBNA sequence analysis. Why was that so important?
Oh, it was absolutely crucial because, unlike a lot of fungal groups, these Archiascomycetes, they don't really have many shared physical features, the kind of things you'd normally use to define a class.
Ah, okay.
So their relationships were just this tangled mess until DNA sequencing sort of came in like a detective, and that molecular data showed they're a really diverse group, but definitely within the phylum Ascomycota.
Right.
But what's really striking is just how varied they are in lifestyle. You've got some that are saprobic, living on dead stuff, like Saitoella in soil, or the famous Schizosaccharomyces. But then, boom, you've got these obligate parasites like Taphrina and Protomyces on plants, and then Pneumocystis, which is a major parasite in mammals. It's not a neat little box.
So looking at them doesn't give many clues, making the DNA absolutely vital, which I guess leads to the really interesting history part, right? The debates.
Oh, absolutely. Huge debates. These organisms are famous for it. For years, different ones were just misclassified, sometimes put with protozoa, sometimes even stuck in completely different fungal groups like the Mycota.
Wow.
Yeah, like Taphrina. People argued, is it a primitive Ascomycete? Is it a Mycete? Maybe related to rust fungi. It was like a constant identity crisis.
And the big one was Pneumocystis.
That's probably the most dramatic example, yeah. For the longest time, decades, the scientific consensus was, it's a protozoan, totally different kingdom of life.
Seriously?
Seriously. Even though there was some weird early evidence about spores, it wasn't until 1988, with really solid DNA sequencing, that it got definitively placed. Fungus. An early-diverging Ascomycete. That discovery it landed like a bombshell, changed everything about how we understood that organism.
So this isn't just scientists tidying up labels. It's deeper, isn't it, about evolution?
Exactly. That's the profound part. This taxonomic fluidity, it's really significant.
Yeah.
We now think the Archiascomycetes might represent some of the earliest branches off the Ascomycete tree.
Okay.
Which makes them incredibly valuable for figuring out the whole evolutionary story of fungi. But here's the catch. Because they are so inconsistent physically, biochemically, and there's some statistical wrinkles with the rDNA analysis, the group Archiascomycetes actually isn't a formally described class. It's still sort of a collection of related but very diverse mavericks, united by genes, not looks.
Got it. Okay, so with that big picture of how complicated they are, let's zoom in. Maybe start with the order Taphrinales. That includes Taphrina and Protomycetaceae. You said these are plant pathogens with a split personality.
Yeah, that's a good way to put it. And their impact, ecologically and agriculturally, is pretty significant.
How?
Well, basically, everything in this order is a parasite on angiosperms flowering plants. And they cause all sorts of visible problems.
Right.
Galls, weird thickenings, blisters on leaves, lesions, and those really dense twiggy growths called witch's brooms.
Ah, witch's brooms. I've heard of those.
Yeah. And a really key example, one that hits our food supply, is Taphrina deformans. It causes peach and almond leaf curl. It's a big deal economically. It can really strip the leaves off the trees.
Oof.
Other species cause things like plum pockets, or those witch's brooms you see on cherry trees sometimes.
Now, what about this dimorphic thing, their split personality? What does that mean for them?
It means they have two distinct forms they can exist in. It's kind of like a Jekyll and Hyde situation. There's a free-living saprobic stage where they exist as single yeast cells. These are usually haploid one set of chromosomes.
Okay.
And then there's the infective parasitic stage. This is when they form mycelium, those thread-like fungal structures.
Right.
And this stage grows inside the host plant. So really adaptable strategy.
Super clever. So how does this play out in their life cycle? Can you walk us through Taphrina deformans?
Sure. Let's trace it. It starts with the haploid ascospores. Think of them like fungal seeds. These spores land somewhere, and they start budding off smaller, single-nucleus haploid cells called blastospores. These are basically the yeast cells.
Right, the free-living stage.
Exactly. These yeast cells can just hang out on the plant surface. Then, when conditions are right, usually springtime, young plant tissues, they somehow switch gears and start the parasitic mycelial stage. It's thought maybe two yeast cells fuse, but the details are still a bit debated.
Okay.
Anyway, this mycelium grows inside the host, sort of weaving between the plant cells or just under the surface layer, the cuticle. Then as it grows, certain cells in the mycelium enlarge. These become ascogenous cells. And inside these, the critical step happens, karyogamy. The nuclei fuse. Now the cells deploy two sets of chromosomes.
Got it.
These diploid cells then directly elongate and become the little sacs that will hold the spores.
Ah, the spore sacs.
Right. Inside each ascus, you get meiosis, then mitosis.
No.
Long story short, you end up with 8 nuclei that develop into 8 new ascospores. These ascus all form a layer right on the host's surface, pushing up against the plant's cuticle.
Building pressure.
Exactly. Until they just burst through. The spores get shot out, usually just by the tip of the ascus rupturing under pressure. There's no fancy mechanism. And then they disperse, ready to start the whole cycle over.
Wow. It's complex. And you said some parts are still being figured out.
Yeah, like the precise trigger for the mycelial stage, or exactly how the nuclei behave during that transition. Science is always refining the picture.
It really is. So how do the Protomycetaceae differ?
Well, they're generally studied less, mostly because they tend to infect weeds, things without much economic importance. But biologically, they're fascinating for understanding evolution. The key difference in Protomyces is how the spores form. It involves something called a synascus, like a compound ascus. The cytoplasm inside gets divided up before meiosis happens, separating the diploid nuclei into compartments first. It's a neat variation on the theme.
Interesting. Okay, let's switch gears completely from plant parasites to, well, lab heroes, the order Schizosaccharomyces.
Schizosaccharomyces. Fission yeasts. You called them unsung heroes.
I think they often are. Ecologically, yeah, you find them naturally in sugary places, tree slime, honey, fruit juice, that kind of thing. But where they really shine is in the lab.
As model organisms, right?
Exactly. Schizosaccharomyces pombe, often just called S. pombe, is a huge deal in genetics and molecular biology, especially for studying the cell cycle, how cells divide and control that process.
Why them specifically?
Well, partly because its cell cycle timing and control mechanisms are actually a bit more similar to, say, human cells than Baker's yeast, Saccharomyces cerevisiae, is in some ways. So for certain questions, it's the better model.
Ah, interesting. And they have uses beyond basic research, too, biotechnology. They do. There are strains that can produce useful compounds like citrulline. Some can actually convert components of petroleum, like kerosene or diesel, into protein.
Whoa, really?
Yeah. And others are good at breaking down waste from brewing. Plus, there's a neat little lab trick. Schizosaccharomyces octosobris. Its spores contain starch, amylose specifically. Add iodine, they turn blue. Super easy way to show students yeast ascospores.
That is clever. Okay, the name fission yeast tells us something about how they reproduce asexually, right? How's that different?
Very different from budding yeast. In budding, a small daughter cell grows off the side. Fission yeast don't do that. After the nucleus divides, the whole cell gets longer. Then a new cell wall, a septum, grows inward from the sides, right across the middle, slicing the cell neatly into 2 equal daughter cells.
Like cutting it in half.
Pretty much. Very symmetrical. And their sexual reproduction is cool too, especially in the Ascoctosporus. Mainly because the diploid phase is so short. The normal cells you see are haploid, one nucleus. Any one of these cells can basically act as a gamete producer, a gametangium. 2 cells of different mating types just fuse together. Plasmogamy, then karyogamy happens right away, nuclei fuse. And that diploid zygote, it immediately becomes the ascus.
No waiting around?
No, meiosis happens inside it, then another division, making 8 haploid ascospores. Then the ascus wall just dissolves, lets the spores out, and they start new haploid lives. Very quick trip through diploidy.
Fascinating. Okay, last stop on our tour, Two genera that are kind of outliers, not formally in orders within the Archiascomycetes, Saitoella and Pneumocystis.
Right, two very distinct characters. Let's take Saitoella first, Saitoella complicata. It's an asexual yeast, first found back in '67 in soil from the Himalayas.
Okay.
And the interesting thing is, initially, researchers thought it was a Basidiomycetes yeast, a different major fungal group altogether.
Another case of mistaken identity.
Exactly. And this is where you really see the power of newer techniques. Things like protein analysis, electron microscopy, DNA comparisons, and especially that our DNA sequencing we talked about. They all pointed towards it actually being an early branching Ascomycete.
So technology corrected the classification.
Precisely. Shows how our understanding grows. But Saitoella is still weird. It only reproduces by something called enteroblastic budding, where the new cell wall starts forming inside the parent before the bud emerges. That's more typical of Basidio yeasts.
Huh. So it has traits of both.
Sort of, yeah. It's classified as an Ascomycete based on DNA, but its budding looks more Basidio, and it's rare. No close relatives known, and apparently it hasn't been found again since that first discovery. Bit of a mystery.
Wow. Okay, and then there's Pneumocystis.
Yeah.
The medical maverick. This is the one with the huge reclassification story, right?
That's the one. Pneumocystis carini, though we now know there are different species specific to different mammals, the one in humans is often called P. giravetii. It's an extracellular parasite, lives in the lungs.
And causes pneumonia.
A very severe, often fatal pneumonia, specifically in people with weakened immune systems. It became widely known during the AIDS epidemic, but it affects other immunocompromised patients too. It binds to lung cells and can sometimes spread.
And the reclassification, that was the protozoan thing.
Yes, that 1988 bombshell. Before that, everyone knew it was a protozoan. The DNA results showing it was clearly a fungus, an early-diverging Ascomycete. It completely changed the game.
How so? What did that change practically?
Well, think about treatment. Suddenly you realize antifungal drugs might work. Research explored things like Benomyl. It also helped make sense of why some existing drugs like pentamidine were effective, because pentamidine, it turns out, also works against some other Ascomycetes. Classification directly impacted medicine. A DNA sequence literally helped save lives by redirecting treatment strategies.
It's incredible. Just changing the category makes that much difference. Can you sketch out its life cycle? Is it as weird as the others?
It has its quirks. It starts with these small haploid cells in the lung alveoli. They probably divide by fission, like Schizosaccharomyces.
Okay.
Then these haploid cells fuse in pairs. Karyokemy happens right away, forming a diploid zygote. The zygote develops into a young ascus with a distinctive two-layered wall.
Right.
Inside, mitosis makes 8 nuclei. But here's a really unique bit, how the spores get packaged. An internal membrane system forms, folding in from the cell surface. But initially, it wraps around all 8 nuclei together.
All at once, that's different.
Very different from Taphrina or baker's yeast, where spores form individually. Only later does this membrane system divide things up to delimit the actual ascospores. Then, the ascus wall breaks, releasing the spores.
But we have to remember, studying this is hard. You can't grow Pneumocystis in a standard lab culture. And in the lungs, the development isn't synchronized. You see all stages at once. Makes it tough to piece together the exact sequence.
Yeah, I can imagine.
And adding another layer, the latest thinking on Pneumocystis pneumonia is shifting. It used to be thought it was mostly reactivation of a dormant infection people picked up earlier in life.
But more recent studies strongly suggest it might actually be caused by constant reinfection from some environmental source we still haven't pinned down.
Wow, so people are getting newly infected again and again.
That seems to be the idea now, which, if true, totally changes how you'd think about preventing it. It's not about managing latency, it's about avoiding exposure. Still lots to learn there.
Man, what a tour through the Archiascomycetes. It's clear this group, largely defined by DNA, just throws curveballs at our standard classification ideas.
They really do.
From Taphrina, the plant pest with its dual life, to Schizosaccharomyces, the super useful lab yeast, and then Pneumocystis, the fungus that masqueraded as a protozoan and changed medicine. Their diversity in looks, lifestyles, impact, it's just huge. Really shows how science is always, redrawing the map.
Absolutely. So what's the big takeaway here? I think it's that discovering and maybe more importantly, reclassifying organisms like these Archiascomycetes.
Yeah.
It shows how our understanding of life itself is constantly evolving.
Yeah.
Each time we shuffle these categories based on new evidence, especially genetic evidence, it's not just about neatness. It deepens our appreciation for just how complex and interconnected biology is. And like we saw with Pneumocystis, these shifts aren't just academic. They can have real, tangible effects on health, on farming, on the environment.
Right.
It proves that even these tiny, sometimes obscure organisms hold massive lessons about life's basic rules. They keep pushing us to ask more questions, to look closer, and to keep learning.
Couldn't have said it better. Well, that wraps up this deep dive. Thanks so much for joining us on this exploration of the Archiascomycetes, the fungal mavericks. Hope you picked up some new insights, maybe had a few aha moments yourself.